the third and final step, the electron transport chain. You might be wondering at this point, okay, Miss Mitchell, where is all the energy? The whole point of this was to break glucose down into energy, and all we've done is make a couple ATPs in glycolysis, a couple ATPs in the Krebs cycle, so that doesn't seem like a lot. Well, you're right. That's what we've been waiting for, is this third step, to make most of our ATP. And how are we going to do it? We're going to use all the energy that we've been stockpiling in that NADH and FADH, the temporary energy storage molecules. So that's where they're going to come in handy. Now this is going to be occurring actually on the inner membrane of the mitochondria. So if you have a little picture of a mitochondria here, you've got your squiggly inner membrane, this is where it's occurring. This side of the blown up gigantic membrane represents the intermembrane space. Okay? That area in between the two membranes. This side is going to be the mitochondrial matrix, so the very, very center. So what we're going to be seeing here is going to be occurring along that inner membrane. This whole thing is a giant blown up version of what's in that circle. Now here's what we're going to do. We're going to take the NADH that we made and the FADH that we made, and we are going to drop off the electrons that we picked up earlier. Okay, these electrons are stored forms of energy. Now that's going to turn NADH back into NAD plus and back into FAD plus. And those are going to actually get recycled and just go back to the previous steps and keep picking up electrons and then coming back here and dropping them off. Now we're dealing with electron exchange, so don't forget this is yet another redox reaction. Leo says, "Grr, who's losing the electrons?" These two, they're being oxidized. Who's gaining the electron? It's actually this molecule here called a proton pump. So that is what's being reduced. And ladies and gentlemen, this electron transport chain of molecules you see here is actually going to do just that. It's going to transport that electron all the way through and out. Now the reason that we do this is it actually is going to drop off some energy to each of these molecules, or proteins, as it goes through. And it's going to allow us to pump protons, just like it says. So guys, there are protons built up inside of the mitochondrial matrix. And every single time that we send an electron through this chain, it allows each proton pump to send a proton to this side. And we start building up a lot of them. We start creating a gradient. And what we have is a high concentration on this side and a low concentration, there we go, low concentration on that side. Guys, we are creating potential energy. We've got a high concentration here, low concentration here. These protons do not like being crowded like that. They want to get as far away from each other as they can, go to equilibrium. And we're going to let them do it, but only in a very specific way so that we can capture the energy as they move to equilibrium. Ladies and gentlemen, this is an ion channel. And we learned last unit that ion channels allow ions to go across the membrane, get through those phospholipids. And we're going to let these protons flow from high to low concentration through the ion channel. Guys, your cells are smart. We attach something on the end called ATPase. Now the A's indicates we've got an enzyme here. And this enzyme's entire job is to turn ADP into ATP. So every time a proton zooms through this ion channel, we're going to be able to turn ADP to ATP with the help of that enzyme. And that's where we start really making the energy from the glucose that we've needed all along. Last little thing, we've got to do something with these electrons and with these protons that have been coming through the ion channel. Some of them are going to be picked up by oxygen. So this little electron here and this little proton here are going to combine with oxygen and give us water. Remember, an electron plus a proton makes a hydrogen atom. Combine that with oxygen and we'll make water. Yes, I know that this equation is not balanced. The numbers don't add up, but that's what will combine together to get our final product. Ladies and gentlemen, oxygen is called the final electron acceptor. And it is really, really important, because if we didn't have this oxygen to pick up these electrons as they came through, the entire process would stop. Everything would get backed up. These wouldn't drop off their electrons, NADH and FADH, which means that they wouldn't be recycled back to NAD plus and FAD plus, which means that the Krebs cycle would stop, glycolysis would stop, and the entire process completely shuts down. So ladies and gentlemen, we've finally made our ATP. Quick review. We take our NADH, FADH, do a redox reaction, drop off our electrons, send them through the electron transport chain, and every time we do it, pump protons from one side of the membrane to the other. That creates a concentration gradient. We let the protons flow down through that gradient, and we harness the energy with our ATPase. That helps us create the ATP we've been looking for. Oxygen will accept the electrons that have gone through and some of these protons to create our final byproduct,